especially mixed populations where you have vaccinated groups, cohorts, unvaccinated cohorts, and cohorts that have had natural infection.
I agree. I think it's particularly for breakthrough infections of understanding, what's going on in those populations as well. Is some new variant popping up in those populations? But you don't need to sequence every single virus, and so I think the sampling approach of doing surveillance, I think, is actually quite reasonable. Sequencing a bunch of Delta variants, it's all Delta, that's fine. I think particularly times like these where you're seeing maybe a variant switch or there's a possibility of a new variant being introduced to the population, is particularly when the most useful times to really do sequencing to understand what's happening, and particularly the difference in the vaccinated and those with prior infection.
Got it. And then kind of back to Max's question. So it sounds like next year you would envision, just given the need for these antivirals for the rapid testing, that you would think there'd be a bigger shift towards rapid versus PCR next year because of that. So would you think that PCR testing, knowing what we know today about Omicron or whatever other types of variants may emerge, would you think PCR testing next year, just would be your guess, would go down in 22 versus 21, and will rapids maybe go up, or do you think PCR could even be sustained at these levels?
And PCR does have some certain advantages, I think, particularly on a health system scale. As you run these in 96-well plates or 384 plates, you can run these sort of mass, fairly inexpensively from a low labor perspective cost. The lateral flow assays are great for the individual patient. It takes 10, 15 minutes. But if you're going to run 1,000 of those, that's hugely labor intensive, and so that is not really practical from a large scale system. But it's great for a doctor's office where you're going to say, let's do this test, boom, we're going to get the result back in 20 minutes, and I'm going to prescribe you drug X, or we're going to watch. And so I think that in the clinic sort of health, doctor's office, clinic sort of perspective, it's great. From a broader health system, PCR still has a lot of advantages from just economies of scale.
Mm-hmm. Sorry, go ahead, Dr. Barra.
I just said I agree completely.
From a manufacturing basis, I joined late, so I wasn't sure your view on whether or not a next-gen multivalent vaccine would be needed, or if they can just adapt the existing vaccines. But I'm just wondering, from a manufacturing basis, is there anything from Omicron in terms of what the vaccine adaptation or would be needed to meet this new variant? Would that create any manufacturing issues, do you think, or is it would the process and the raw materials needed would be kind of very, very similar enough that it's not going to create any bottlenecks from what we've seen thus far?
Yeah, I don't think the mRNA can be tweaked immediately and very easily. I think the biggest question is the regulatory pathway, as far as how much data, what's needed for a regulatory approval.
I would agree. I mean, the rumors on the variant started coming out, I guess, toward the last week sometime. If companies like Moderna haven't already started synthesizing the new spike, I'd be surprised. And I'd also potentially be surprised if they didn't have it by Tuesday or Wednesday of this week.
Got it. Okay, great. Maybe with that, I'll turn it over to Steve.
Yeah. And I think Yaron has an email question.
The email question is, what do we know about, I think it goes back to our original discussion, on some of the new mutations related to the spike protein and Omicron. How can we make any predictions prior to incubating this variant in human serum? Or to your point, we need to wait until mid-next week to really understand the intent.
I agree. We know that Omicron is a Greek letter. Everything else is really conjecture, and we need some more data. What are the clinical outcomes? Viral loads in people. What's the particularly neutralizing activity in prior titers? And that's a screen. That's not the end-all be-all, because we do have B cells. They make more antibodies in humans. But that's certainly a starting point to see how big of effect this is. For all the monoclonals, the same thing. What happens with the neutralizing titers? So all those companies, I'm sure, are working on this same thing as well.
Jesse Bloom's group at University of Washington has done systematic mutagenesis across the spike, including all of the variations that have been seen, but also just querying what the role of virtually every position is in how it affects polyclonal sera. And when you look at his blogs, there's about 7 mutations here totally that have large to moderate effects on neutralizing titer, either individually or in combination. So I think we're going to see the most significant shift yet in reduction in neutralizing titers with the Omicron variant that has been described.
And Steve, some of the other questions relating to the impact of Omicron on the orals, and I know you're moving to the orals next anyway. So
Okay, thank you, Yaron. So let's spend a few minutes on the oral antivirals. So since our last call, Dr. Boulware, we learned 2 things about molnupiravir. We got the second group of patient data from MOVe-OUT, and we also saw the FDA briefing documents. 3 weeks ago, you were not very excited about molnupiravir. How has your view changed, and what did you think of the additional data we got last Friday?
Well, I guess I was, molnupiravir I think is still sort of 3rd or 4th line in sort of my pecking order of really therapies. The first is the Paxlovid, the second is monoclonal antibodies. The monoclonal antibodies are going to be the most susceptible to changes and binding, and so that may move down if Omicron becomes more active. And then sort of the 30% effect of the molnupiravir is basically the same as fluvoxamine. Fluvoxamine is an anti-inflammatory, works on host side, has a basic 30% effect similar to Merck's drug, and it's $5 versus $700. And so that's kind of my pecking order. So the 30% effect, so it's kind of borderline statistically significant, and but the question for the FDA, the EUA, is really, is it better than placebo? So not is it the best drug or is it the best therapy, but is it better than placebo? And so in that sense, I still perceive getting an EUA and it being an option. And so while the Paxlovid is certainly a better drug, it's not available today. And so if you can teleport yourself into the future, that's great. But for today, for those that can't get monoclonal antibodies or don't want to get them, having an orally available therapy that has a 30% reduction certainly is beneficial over 0% reduction.
And Dr. Barrett, your view of molnupiravir?
I think the key thing that you need to compare when you look across oral-based drugs is the time of delivery. And if I remember correctly, the molnupiravir studies used patients that were that had been diagnosed up to 5 days earlier versus Pfizer, which mostly focused on people 3 days after diagnosis. And then there's variation in terms of how the diagnostics were done. So I'm less convinced that there's a big difference between the 2 drugs, but I think both drugs again have the potential to make a difference in people's lives.
And Dr. Barrett, your views of the safety profile of molnupiravir?
I worked extensively on molnupiravir, so I think the documents will be reviewed by the FDA and they will make a determination.
Okay. It sounds like you might not be as concerned about the safety profile as people like me who just read documents. Might that be a reasonable conclusion or not?
Again, I'm a little reluctant to answer questions on molnupiravir.
Okay,
unless it's out in the public space.
Okay.
And so, yes, I just would prefer not to comment.
I think the FDA briefing documents for the November 30th meeting actually have pretty good perspective on what the FDA's viewpoint is. And so the FDA does not seem to be concerned about the safety profile. I think individual prescribing doctors, there's still a little bit of questions in the back of your mind. But the FDA seems to be pretty satisfied with the safety data from what they wrote in the documents and how they wrote it.
Okay. And on Paxlovid, what do we know or what can we conjecture about its activity against the Omicron variant, Dr. Boulware?
So the target, that the main sort of protease is conserved across multiple coronaviruses, and so as a conserved protein, it's the same protein. It doesn't vary. That this suggests that there's probably a fairly high. There's a reason why there's not a lot of variation in this protein, genetic variation in this protein, and that it's a very efficient protein. So I don't expect there to be a lot of variation. It's not a